So I'm going to talk about uh, protein synthesis. So the process is always going to go from DNA to RNA to protein. That is central dogma. When you have DNA and it replicates itself, it will always make more DNA. When you go from DNA to RNA, that is transcription. When you go from RNA to protein, that is translation. So there are three different types of RNA. So there is messenger RNA, transfer RNA, ribosomal RNA. Messenger RNA is the DNA sequence that is being read. So it's read from DNA converted into RNA. Transfer RNA brings in the amino acid that will make the protein itself, putting them together. And then the ribosomal RNA is the ribosome is the powerhouse basically, or the factory that puts the proteins together. So when you have DNA, you have guanine and cytosine with 3 bonds, hydrogen bonds. Then you have adenine and thymine with 2 bonds, hydrogen bonds. For RNA, guanine and cytosine have 3 hydrogen bonds. Adenine and uracil have 2 bonds. So for DNA, it's always going to be thymine. For RNA, it's always going to be uracil. So when you read your exam, there's always going to be a set of 3 codons. So a codon is 3 amino acid, 3 nucleotides. I basically told you that those names are basically interchangeable. Nitrogenous base. All right, so those are the common names for them. And then you always have to be able to start the process. So to start the process, the first amino acid will always be a met. For the exam, this is what you will basically be writing, the three-letter name. All right. So for met, you have an A for the first position letter. Then for the second position, you have a U. And then for the third position, you have a G. So when you go over here, you have a met. For the stop codons, it would be a U, an A, and then another A. The other one would be a U, an A, and a G. And then the last one would be a U, a G, and an A. So those are the 3 that I have written right there. All right. Start codon tells you when to start. Stop codon basically tells you enough is enough, like this is the end of the protein. All right. So when you're reading the normal DNA, I'm going to be following this that is in lecture 9. All right. So when you have normal DNA, the messenger RNA is always going to be the opposite of what is what it is attached to. So when you have an adenine, it's always going to go with a uracil. So you will have 3 U's here. You also have a U here. When you have a T, it connects with an A. For the guanine, it is a cytosine. For the cytosine, it is a guanine. For the 2-thymine, A, G, A, G. All right. When you have the anticodon, it is reading the opposite of what this is to bring in the appropriate amino acid. All right. So let me bring this over. So for your protein assignment, you had the transfer RNA. So the transfer RNA for this is going to be A. This is the anticodon that will bring in the appropriate protein. For this one, it's going to be an A, a U, because remember, there's no T in any of the RNAs. Then a G, a C, a U, and then a C, a U, a C. All right. So when you're figuring out what the protein is, you're using this. All right. The anticodon is just bringing the appropriate amino acid over. Hopefully you guys can see that. Okay. So when we're reading the messenger RNA, the first one is U, U, U. All right. So if we look at this, we have a U, we have another U up here, and then we'll look at the U from right across. It is a Phi. So it will be P-H-E. The next one is UAC. We have a U, we have an A, we have a C, it's called Chura. The next one is a GAA. So we go all the way down, G to the A, to the A, it is a glue. And then the last one is a GAG. So G, A, G would be another glue. This is a U. All right. So if you look at it, that is the same sequence that I got. All right. Never go off the transfer RNA. The transfer RNA is the anticodon. So in order to bring in the appropriate protein, it needs to read what the opposite of the messenger RNA is.